Okay. Well, I'm going to talk about the building frameworks that operationalize exposomics and precision environmental health. But this is in the context of, I thought I'd take the opportunity to tell you a little bit more about NIEHS, the institute that I'm the director of. You're going to hear, you heard yesterday about the National Institute on Aging, and you're going to hear more on the NINDS, the American Cancer Institute of Neurological Diseases and Stroke. It's best that you maybe know about NIEHS because you get money from us. But if you're not aware of NIEHS, let me just take a couple of minutes about that. So we are one of the 27 institutes and centers that comprise the National Institutes of Health. And unlike the 26 other institute and centers that are generally headquartered in the greater Washington, D.C., Baltimore area, we're located in central North Carolina. So if you're interested in knowing why we're in North Carolina, ask me at the coffee break. There's a good scientific reason for that and a good political reason for that. So like many of the other institutes and centers, we have intramural laboratories. We have about 55 different investigators studying basic mechanisms of how human biology works and better understanding how different environmental perturbations can be interfering with those biological processes. We also have the second intramural program that relates to our collaborations with the CDC and the FDA for the National Toxicology Program. But the bulk, well, we're not the biggest, we're not the smallest. Actually, we're a bit smaller than the National Institute on Aging. Our budget is about $1 billion. So the key thing is to figure out how can we invest that $1 billion in a way that is most strategic to advance the causes of the environmental health sciences community. So the mission of NIEHS is shown here. It's really all about how the environment affects people in order to promote healthier lives. And our vision is about promoting the global leadership for innovative research that improves public health by preventing disease and disability. So I think the preventing part here is a real differentiator. So many of the other institutes, say National Institute on Aging or National Cancer Institute, focused on how do we cure cancer, how do we cure Alzheimer's once it happens. We're all about trying to develop the strategies to identify, are there environmental variables that may be predisposing an individual to cancer, Alzheimer's, and other neurological diseases, and can we prevent the exposure to those environmental perturbations. So we're just finishing the process of our strategic planning, trying to lay out the strategy for the next 5 years. And I don't have time to go into all of the details, but this is a summary. There are 6 different areas of scientific focus. We'll be polishing this up, getting it out for public comment. So hopefully you'll be seeing this in the next week or two. And then once we have public comments, we will publish this. And again, this will set the stage and help us to better understand how we use that $1 billion every year to support research in the public health community. Now, one of the things, just from my personal standpoint, and part of the reason I'm a genetic and genomics person by training, but I've more recently come to appreciate that it's not just genetics and genomics, and it's not just the genes we inherit from our parents. The environment plays a critically important role in directing both health and disease. So it's got to be a combination of both of these things. So I am personally passionate about working together with the 26 other institutes and centers of the NIH and better integrating environmental exposures, and hopefully in an exposomics framework, integrating this into the way that we study the etiology of human disease. So it has to be a combination of both these things. And in many instances, it's going to be gene by environment effects. You need to develop the strategies to do that. So there are 6 different areas of focus. I'll talk about the exposome precision environmental health today, but we're also very interested in mechanistic and translational biology and toxicology. It's no longer a question of just taking outbred rats and shooting them up with different concentrations of a different chemical and counting lumps and bumps and deaths over the course of 2 years. You know, it's our standard 2-year bioassay. But we want to focus more on mechanistically. How are these environmental chemicals, or how are different social determinants of health actually influencing the molecular biology of the organism? We also have a big focus on climate change and health. Again, I don't have time to get into that. But if you're interested, NIH.gov/climate and health. We have a whole initiative outlined on that website. NIEHS has had a big focus on environmental justice and health disparities. So let's take a look at our strategic plan, and we will continue to focus on better understanding the differential impacts of environmental contaminants on certain populations. And of course, there's a big focus on computational biology and data science. You know, the AI era is here, and we are doing a lot of planning to figure out how do we manage all of the databases that are going to be necessary to make all of the rest of the science that we support happen. So let me get into the details now about the exposome. So I'm going to cross-reference, as I often do. Whereas Gary and Roll's paper in Science, 4 years ago now, wasn't it? 2020. So they really defined and provided really an excellent summary of the exposome. So this was, the exposome concept was originally offered up by Chris Weil back in 2005. And I think, as I understand it, Chris was thinking, well, the Genome Project just finished. They just published the draft sequence of the human genome. And in 2005, we were beginning to appreciate how the Genome Project was really transforming the way that we do genetics and genomics. It really kind of facilitated genomic science in very powerful ways. And it was all about, the Genome Project was all about getting beyond one gene at a time. So if we truly want to understand the molecular biology, we have to get beyond one gene at a time and study the whole genome and study all of the genes at a time. So the Genome Project gave us those incredible tools and capabilities to do that. So, but from the standpoint of the environment, if we truly want to understand the environment, we have to get beyond one exposure at a time. Actually, I think Joel's talk yesterday, when you just break down the air pollution into all the different components, it's actually quite revealing. So if we want to understand all these different things, we have to get beyond one exposure at a time. So one of the things that Gary and Lowell pointed out in their review article is that, in the lower panel here, we want to understand the environment. It's not just about air pollution. It's not just about a pesticide. We have to understand, develop a framework, a research framework to study all of these different things, the physical and chemical exposures. But I think a very important point that they made, and I think it's one that I want to emphasize, because if there's any ambiguity, and I wanted to eliminate that ambiguity, it's not just about the physical chemical exposures. It's not just about air pollution. It's not just about a pesticide. We have to be factoring into the exposome framework social, ecosystem, and lifestyle variables. Diet is really important. Actually, we heard yesterday from in certain experiments where it could be the omega-3 fatty acids in some of the diet that where the association actually goes away. So we've got to be factoring that into the equation. We have to be talking about and factoring psychosocial stress. I think we all of us in the biomedical research enterprise understand psychosocial stress is terribly damaging to human biology. So it can't be just about pesticides and air pollution and other things, but you've got to incorporate psychosocial stress, sleeping, behavior, diet, the built environment. We talked yesterday about the green space. So we need a research framework that brings all of these things together. And it's important, I think, at least from my point of view, because you're factoring the social, ecosystem, and lifestyle variables. There may be dietary influences that can mitigate some of the negative effects associated with physical and chemical exposures. So we've got to develop this research framework that incorporates all of these different things. And furthermore, it shouldn't be just about evaluating what's in the environment. We should also be thinking about how does the human body respond to these different complex environmental exposures. So an area of, say, of epigenetics, actually it's work that we do in my own laboratory. Are there epigenetic signatures of complex environmental exposures? Because ultimately, when your genes start misbehaving, that's what's ultimately going to cause your biological consequences. So the exposome is about all of this, but it's a big task. And I think one of the key things that Roland and Gary and many others have really been pointing out over the past several years is it's lots of different things. How do we do studies that use an exposome framework? We really need an operational definition. What data do we collect? Where do we share the data that we collect in a way that there's seamless integration across laboratories across the US, across the global communities? Now, in some cases, we may need new technologies. So by reference to the Genome Project, just like back in the 1980s when they proposed the Genome Project, I still remember as a graduate student at the time, I'm thinking, what are these people smoking? I mean, they're suggesting we're doing what? We had this big, bold project to completely sequence the human genome at nucleotide sequence level. I mean, come on. I mean, that's never going to happen. But over the course of the last 30 years, I mean, there are technological developments that have happened. And we can do things today in genetics and genomics that we would have never imagined we'd be able to do back in the 1980s when the Genome Project was first launched, when that bold vision was first promoted. So we made new technologies to actually capture the exposome. And I'm thinking specifically around some of these social determinants of health. We can either do questionnaires, or is there ability to actually go in and look at, say, epigenetic marks on certain, say, stress response genes. I mean, there's data to suggest that actually happens. So what technologies are we ultimately going to need to really capture the totality of exposure? And I think I don't know, I first heard it from Gary, but it may have been from others, about, well, let's not wait until we have all of the technologies developed, just like with the Genome Project. They started with some pretty rudimentary tools. I don't know if anyone remembers those cDNAs that you spotted on microscope slides, and you could probably look at 100 genes at a time. Pretty rudimentary compared to the RNA-seq technologies that we have today. Well, they started with that, and we can start with some of the tools that we have available now. So we have some pretty awesome capabilities to do high-resolution mass spectrometry. It may not be as high throughput and as cost-effective to do on a large scale, but let's start with what we can do. So again, modeling on this, what we do with the genomics community. Let's start with a bold vision. Okay, we have, and I think we have the bold vision, the exposomics strategies on capturing environmental data. And then let's start with the things that we can do now, with the understanding that if we truly want to capture and we truly want to enable this bold vision, let's develop new technologies that will improve the cost effectiveness and the throughput of our ability to capture data. So one of the challenges now is that, and actually Gary was here and organized a meeting at the Banbury Center. I think it was at the beginning of December. In fact, during the question and answer, maybe Gary would be willing to take a few questions and ask a few things. But this was all really about, let's get some people together and let's start talking about this bold vision. And what are some of the things that we can be doing to operationalize the exposomics framework? So that as a result of that meeting, again, it's just summarizing some of the information that Gary's provided to me. It was the first effort was to define what is the exposome. So the exposome is the integrated compilation of all the physical, chemical, biological, and psychosocial influences that impact biology. You know, it emphasizes not just the chemical, physical, and chemical exposures. It's all the things that I showed you on that first slide. Okay, we have a common definition. It's a transdisciplinary field aimed at a discovery-based understanding of how the exposome influences biology and health. And then you have I think emerging from the conference was the operational definition. It's really about integrating data from a variety of interdisciplinary methodologies and streams to enable discovery-based analyses. So I think that's really a key summary point here. And also coming out of the meeting is it's not a simple implementation. It's very unlikely that anyone, I mean, Joel's pretty talented, but I doubt that you can actually go in and do the totality of exposures over the life course. It could take a little bit more computational capability than you have right now. But can we actually take the data from multiple different studies? Okay, and start integrating them together in a data infrastructure so that Joel can share his data with Gary and with Bob and with others, and so we can all benefit from each other's work. But I think the key thing here is, and again, Gary can take questions on this, I think it's important that the exposomics is different than environmental health studies. I mean, there's nothing wrong with the environmental health study where you study only one component, PM2.5. I mean, that needs to continue to be done, just like genetics and genomics. Genomics is studying the totality of exposures. Genetics is studying, in many cases, one gene and different alleles of the gene. And they both need to be done, and they're both worthy to be done. So it's an important thing to just keep in mind. So if you operationalize exposomics, you're going to need tools to capture the external exposome. You know, the macro scale. We had a fantastic GIS workshop on whatever day it was it yesterday? Time just moved so quickly. But there's just incredibly powerful things you can learn about environmental exposures by knowing where someone lives, where they work, where they've lived, or where they've worked in the past. So we need to develop those types of tools. Also wearable sensors, some of the remarkable the wristbands for capturing volatile organic compounds. So we need to be thinking about these things. Also internal exposure and biological responses. I mean, we do have capabilities to look at the high resolution mass spec. We learned a little bit yesterday about some incredibly powerful tools around analysis of urine. So let's bring some of these things together. And as well as to capture metabolomics, other omics, proteomics, adductomics, looking at different changes in the DNA as a consequence of exposure to different complex environmental mixtures. And also, importantly, the biological responses. So what are the epigenetic changes that occur as a consequence of the environmental exposures? I'll show you some data in just a few minutes from my own lab about the environmental exposures that occur during pregnancy and how that can be affecting how your genes are working much later in life. So it's this whole concept of that. So we've got to pull these things together. Of course, we have the HERE resource, and this, just quickly summarizing here, it offers the hypothesis for the exploratory analysis, kind of getting beyond one exposure at a time, but looking across. And this is the kind of the pragmatic exposome. This is stuff we can do right now with the existing technologies using, say, NMR and mass spectrometry. But I think the future is really, let's pull together some additional planning efforts. I mean, Danbury was a great start, but let's actually, so the solution that we've come up with is let's charge a center, a single center, not multiple centers that compete with each other, but a single center that will be reaching out to the global environmental health sciences community, and actually a center that is in charge with developing a conceptual framework to address exposomics, promoting what type of technologies do we need to be developing to actually achieve that bold vision of doing the exposome, human exposome. And most importantly, build the exposome research community that fosters national and international collaborations. So I think that's where the Genome Project really blew it in the beginning. You had all of the different countries, you have different labs within the United States, these different sequencing centers, and the Broad was competing with the Baylor, and they're competing with WashU, and it was going nowhere. And just really to Francis Collins' credit, he actually pulled them together and said, look, we need to start collaborating together and working together. We had little influence from Craig Venter, but nevertheless, pulling people together on an international scale actually made the Genome Project work. So let's do it differently with the exposomics. Let's get people working together around a common set of goals. So we hope to be the proposals are in. I see many of you in the audience that have a great interest in this. So we'll be announcing who will be awarded this contract soon. So probably in the July timeframe. So we're not the only ones doing this. If you're not aware of this, there's the IHEM. It's the International Human Exposome Network. I first learned about this from some of our colleagues in Europe. But there were many countries across Europe that are engaged in discussing really the purposes about improving global research and cooperation on the exposome. And so once we have our center awarded, we were hoping that the PI of that center would become part of this IHEM network. So working on a global scale to develop common approaches to studying the exposome. Now the IHEM had a kickoff meeting. It was back on February 27th, 28th. It was in Brussels. We attended by virtually using good old Zoom sessions. And it was really all about designing a global exposome network, identifying and making available exposome tools and resources on a global scale. So to evaluate the proposed network and to and its toolbox by a series of case studies. Can we come up with individual examples where we can better understand how this is all coming together? And develop a roadmap for future exposome research, not just across Europe, but across the globe. And engage stakeholders. So getting the entire community working collaboratively together. I suspect there's going to be some cultural changes. A lot of people in biomedical science, we're not used to seamlessly sharing our data and working collaboratively together. So part of our challenge at the NIH is to create the funding mechanisms that promote collaborative activities. So that's the exposome. And so let me spend just a few minutes now on talking about this, the precision environmental health. So what is it? I mean, the first time I heard about this concept was actually when some of our grantees and prominent members of the environmental health sciences community gave a presentation at one of our council meetings. So Cheryl Walker, Andrew Baccarelli, and Dana Doyle and I talked about this concept of precision environmental health. And this is something actually personally I'm very interested in, because this is all about kind of getting beyond population-based risk assessment. So you can see on the right, we not only look different, but we are biologically very different. And we respond to environmental exposures in different ways. So can we then use some of the powerful tools of, say, genetics and genomics, as well as other strategies around the exposomics framework, begin to capture environmental exposures and then cross-reference that with individual biological variability. So this complements the precision medicine program. So many of you may have heard of precision medicine, and that's all about sequencing someone's genome. And then once the thought was, amongst the genetics and genomics folks, if you sequence someone's genome, you'll know everything you need to know about their propensity to develop disease and their therapeutic response and everything else. Well, we all know in the environmental health sciences community that genetics plays a part of this. The genetic background is important, but it's not everything. And we need to be factoring in, say, some of the epigenetic changes and other biological variability that's associated with environmental exposures. So it complements precision medicine, but precision environmental health is all about G by E by D. So the G is about genetics, the E is about environment and epigenetics, and then the D, as you can imagine, there's a lot of data management in this. So if you're now talking about developing huge data repositories around exposomics and exposomics framework, and you're now crossing that with whole genome sequence from hundreds of thousands of individuals. Joel, that will keep your computers churning. So we need to develop the data management strategies to really pull all this together. Just take 1 second. This is really, so we've got the diet Pepsi, it's not food. Okay. But it's wet for the moment. So the gene part of this is complicated. And it's complicated because there's no single gene that is responsible for dictating how an individual responds to the environment differently from another individual. So we know that there are genetic variables. In fact, I don't have time today, but I can show you data using different inbred strains of mice. There can be very profound genetic background impacts on the response to benzene or the response to different types of drugs. But it's complicated because the genetic propensity here falls into this category of the complex trait. So this is interesting to the genetics and genomics of the biomedical community because most genetic heritability associated with, say, type 2 diabetes and obesity, they're complex traits. In fact, human height is probably the winner as it relates to the complex trait because there are over thousands of genes of sequence variations from that you inherit from your parents that will dictate how tall an individual will be. So all of these different gene variants are working collaboratively together. It's a genetic feature, but they work collaboratively together to ultimately dictate what the phenotype or what the outcome will be. So that's true for environmental exposures. So how do we do this? So it was back in September 2019, Francis Collins, who was the director of the NIH at the time, pulled together a group of genetics and genomics experts from across the NIH and across the global community. And that meeting was I attended on the part of NIHS. And the idea was, well, how do we kind of embrace the challenges associated with complex traits? Because if we truly want to understand potentially hundreds of different gene variants associated with common diseases, we're going to need cohorts of tens of millions of people. Okay, so how do we do that? That's a big task. Does every country, does every organization have to assemble a cohort of tens of millions? And the answer is no. So if you look across the globe, there are big genetics and genomics efforts. In many cases, there are millions of people that are part of these cohorts. So the idea here is, why don't we work together? So why don't we collect data on the cohorts that we're studying in Northern Europe, in Iceland, in the United States, in Japan. And if we collect data that can be seamlessly integrated together, we can all benefit from each other's work. So this conference was all about establishing something called the International Common Disease Alliance. It's the ICDA. And it's really the commitment on the part of these different large cohorts doing genetics and genomics to essentially develop the standardized ways of collecting the data and then making this seamlessly available across international boundaries so we can all benefit from each other's work. Okay, the work we've done in the United States is still done in the US. The work that's done in Iceland is still done there. But if we now have a database infrastructure where these things seamlessly connect with each other, and you don't need a PhD or a postdoc in Joel's lab to figure out how to manage all this stuff, it's immediately available to. So there was a commitment to actually undertake the M2M challenge. This was about mapping to mechanisms to medicine. Okay, I thought that was great. Well, I raised my hand, and I said, well, Francis, we just spent the morning talking about all of this fabulous stuff about complex traits. We never mentioned the word environment once. Okay? This is a problem. And he said, well, Rick, how do you study the environment? This was back in 2019. Eric Lander was there as well. I said, well, Francis, let's talk at the coffee break. So we've been talking since 2019 to these people who are involved in the ICDA. And they're never, I mean, my message to them is you're never going to enable precision medicine and to get to where you want to be if you're looking exclusively at genetics. Genetics is important. Genetics and genomics is important, but you have to factor the environment into the equation. Okay? So the good news is that they're listening to our message. In fact, more recently, the NIH contribution to the ICDA is a 1 million person cohort that's sponsored by the NIH. It's actually funded out of the Office of the Director. And it's really all about individualized prevention, treatment care. The new CEO of the All of Us program, Josh Denny, came from Vanderbilt. And he doesn't need to be convinced that the environment is important. In fact, also Jeff Ginsburg, who is a genetics and genomics person, we've had long discussions with Jeff about the importance of the environment. He doesn't need to be convinced that the environment is important. So we've actually been working with them. So they've been collecting genomics data. They're sequencing a million individuals, sequencing their entire genome, collecting transcriptome data, proteomics data. They have wearables to collect mainly clinical data. These great Apple Watches are terrific for capturing lots of clinical data. They have biospecimens, the electronic health record, behavior surveys, physical measurements, and now we're actually adding a piece of the pie, environmental data. The challenge we have is that Josh came and said, okay, Rick, we're fully convinced, okay? What do we do? How can we integrate comprehensive exposomics data into the All of Us cohort? We want to do it on 1 million people, and we want to do it cost effectively, and we want to do it fast. Okay, well, we can't do that overnight, but what we decided is that we would actually undertake this collaboration, integrating exposomics, data collected in an exposomics framework into the All of Us program. We're going to do it in 3 phases. The first phase is remarkably, when they collected the data on these 1 million people, they never asked them where they live or where they work. So the first step is to go in and collect GIS data from the individuals. It's going to cost $30 million. If you go back and do that, they're doing that. So I'm sure that you will be very pleased to hear about this. So that's essentially been done. In fact, very soon, that information is going to be available for the Environmental Health Sciences Committee to start mining. The next phase is to develop additional tools. We heard yesterday this fabulous workshop about developing the tools to connect GIS coordinates to environmental exposures. And there are a lot of complications on this, but nevertheless, let's build those tools so you can connect these things together. And I'll talk about phase 3 here in just a couple of minutes. But once you have this, of course, what we heard yesterday, Once you know the GIS coordinates, there are phenomenal global air quality models. In fact, you map the entire, if you weren't there yesterday, you really missed a great workshop. But the entire globe is mapped into little coordinates. And so you know exactly what the air quality is in different parts of the world at any given time. And there are longitudinal measures that you want to be able to tap into. So you can now know what happened if you were in New York City last July as a consequence of those Canadian wildfires. That can now be part of your environmental exposure data if you put all these things together. Daily weather, daily weather, and of course the CDC Social Vulnerability Index is a step in the direction of integrating some of the social determinants of health into all of this program. So once you have this data, then you begin to study those gene by environment effects. So the phase 3 is really going beyond just GIS data. So it's about whole genome sequencing, electronic health records, extensive surveys on lifestyle and social determinants of health, thorough analysis of biospecimens, doing the high resolution aspect, the things that we can do within the HERE program today. So we've decided that we're not going to do it on 1 million people. We can't afford to do that. But let's take a relatively small cohort of about 1,500 individuals, type 2 diabetics, 1,500 controls from the All of Us platform, and then do the biospecimens, send them off to the HERE resource, do the whole genome sequencing. And what we can do is, type 2 diabetes is great because we know a lot about the complex traits associated with type 2 diabetes. So we can calculate the polygenic risk scores. But we know just calculating polygenic risk scores doesn't give us everything we need to know about predispositions to type 2 diabetes. But now we're hoping that by adding the additional environmental, the exposomics data, we can calculate essentially the equivalent of a polygenic risk score, a poly-environmental risk score, and then blend the two of these together, and we're hoping that this would be kind of a pilot to really bring to everyone's attention the importance of capturing the environment. So anyway, just a few details here. Lots of different things that we're proposing to analyze. And in the end, it's really about combining the genomic data, questionnaire data, the GIS-based exposures, bringing social determinants of health, the electronic health record data, integrating all of this together and developing those computational tools that bring all of these data sets together to ultimately better understand individual risks. So just since this is an aging meeting, I thought I would just take a couple of minutes reminding everyone of the hallmarks of aging, and many investigators, many of them supported, many of you here, supported by the National Institute on Aging, come to appreciate that there are many different hallmarks. There's genomic instability, telomere attribution, telomeres shorten or lengthen over the course of time. In fact, we work together with the National Institute on Aging to standardize the ways that you capture telomere length. Remarkably, there are all sorts of different technologies that are out there, and they don't necessarily correspond to each other. So it's just an example of how you really have to work on standardizing the way you do things. But there are also epigenetic alterations. In fact, there's a set of methylation loci in the genome that you can calculate the biological age of an individual. And you can compare that to the chronological age. There are mitochondrial dysfunctions. So lots of different things that are happening. The big question is, how many of these things are influenced? What's the role of the environment? Can we bring an exposomics framework to studying the hallmarks of aging? So we know, just real quickly, telomere length can be influenced by single exposures that have been studied. Cadmium, psychosocial stress can shorten the telomeres. But the other thing we understand is that there's a program, a gene expression program throughout the life course. Okay, and this is dictated by epigenetic modifications, methylation changes in the genome. And we also know that there are periods, there are windows from this developmental program that can be influenced by environmental exposures. So this is just an example of work from my own laboratory here. So we treated, well, first of all, just looking at, we did a whole genome methylation analysis. This would be this is in the liver of a mouse. And you can see here, these are hypomethylation in yellow. And there are different loci that have different degrees of methylation. And that changes over the life course. So at day 22, when the animals are born, we have a pattern of methylation. At 6 months, 12 months, and 18 months, it changes. And what we did in this case is we treated mothers with a pesticide. Many of you are familiar with this. It's rotenone. It actually targets complex 1 in the mitochondria. And you can see here, lots of genes are not changed, but there are many different genes where that developmental, that age-related methylation profile is altered as a consequence of the exposure with rotenone. And the rotenone exposure occurred only during development. It stopped at birth. So things that are happening at 18 months. There are lots of different genes here whose methylation status has changed at 18 months. So there are lots of different things that are happening. We've sponsored the Target 2, which is looking at epigenetic modifications in different cell types and looking to see whether, say, peripheral blood lymphocytes are accurate reporters of some of the epigenetic changes that perhaps happen in the brain. So lots of different things happening, lots of challenges as we go forward. And in the interest of time, let me just emphasize one of the last ones here. It's the collaboration. To make all this happen, we're going to need collaboration on a global scale, an unprecedented degree of collaboration, where we bring people with complementary skill sets working together to achieve common goals. So with that, I see that I'm right at time, so thank you for the opportunity to share some ideas, to take a look at our strategic plan, and feel free to give us some feedback.